The overall goal of the experimental technology demonstrated here is to screen compounds against growth and proliferation of intracellular amastigote stage of Leishmania donovani, the causative agent for visceral leishmaniasis, with a primary objective to discover new drugs for treatment of leishmaniasis. First, THP1 human acute monocytic leukemia cells are differentiated in vitro into adherent macrophages. The differentiated THP1 cells are then infected with promastigotes of the Leishmania donovani parasites, which allows invasion of the promastigotes into the THP1 macrophages, their transformation into amastigotes, and intracellular proliferation. Next, the Leishmania-infected THP1 macrophages are exposed to test compounds and standard drugs, and then undergo controlled lysis, rescue of amastigotes, transformation of residual amastigotes to promastigotes, and quantification of promastigotes by colorimetric cell growth assays. Results are obtained that show the anti-Leishmania effect of test compounds and standard drugs on intracellular growth and proliferation based on quantification of intracellular amastigotes.
Leishmaniasis is a major global health threat, especially in tropical areas of the world. New drugs with better therapeutic profiles to treat this disease are urgently needed. We at the National Center for Natural Products Research have launched a multi-directional program for the discovery of new treatments for tropical parasitic diseases.
Unlike reporter gene and microscopic image analysis assays, which may not differentiate between the live and dead amastigotes, this assay measures only the live intracellular amastigotes. We have optimized a parasite rescue and transformation assay with differentiated THP1 cells infected in vitro with the Leishmania donovani for screening the pure compound and natural product extracts and determination of efficacy against the intracellular Leishmania amastigotes. The experimental details of this assay will be demonstrated by Mr. Surendra Jain, who is the research and development biologist in my lab and routinely performs this assay, and Mr. Rajneesh Sahu, who is also working as a research and development biologist in my lab.
There are 4 basic steps in the assay. Step 1 is seeding and terminal differentiation of THP1 cells. Step 2 is in vitro infection of differentiated THP1 cells with Leishmania promastigotes. Step 3 is treatment of Leishmania-infected macrophages with the test compounds or standard drugs. And Step 4 is quantification of the growth and proliferation of Leishmania parasite amastigotes with microscopic image analysis or parasite rescue and transformation assays. Harvest a 4-day-old culture of THP1 macrophages and prepare a cell suspension in rPMI supplemented with 10% heat-inactivated FPS with a concentration of 2.5 times 10 to the 5 cells per milliliter. Add 25 nanograms per milliliter of PMA to the cells to trigger differentiation. Then, transfer 200 microliters of cell suspension to each well of a 96-well plate and each chamber of a 16-chamber glass slide. Once the cells have been plated, place the slide and the 96-well plate in a 37°C incubator and incubate the cells overnight to allow nearly complete cellular differentiation to occur. For the infection, use a 5 to 6 day old promastigote parasite culture in which most organisms are in the infective metacyclic stage. Unlike the procyclic stage promastigotes, which are short ellipsoid cells with a short flagellum, weakly motile and often found in rosettes, the infective metacyclic organisms can be recognized by their long cylindrical shape and high motility. Harvest the parasites and prepare a promastigote suspension of 2.5 times 10 to the 6 parasites per milliliter in 2% FPS rPMI. Retrieve the differentiated THP1 cell cultures from the incubator. Processing the 96-well plate and glass slide in parallel, remove the supernatant from the cultures. Then, wash the cells by adding pre-warmed serum-free medium. Remove the serum-free medium and replace it with 200 microliters per well of promastigote suspension, achieving a ratio of 10 parasites for each THP1 cell. For control wells, add 200 microliters of 2% FPS RPMI medium instead of the parasite suspension. After adding the parasites, incubate the co-cultures at 37 degrees Celsius for 24 hours to allow the infection to occur. From stock solutions of the drugs to be tested, prepare 6 serial 1:5 dilutions in 2% FPS RPMI, such that the concentrations of each dilution is twice the intended final test concentration. As controls, standard anti-leishmanial drugs such as amphotericin B, pentamidine, miltefosine, and sodium stibogluconate may be used. Before adding the drugs to the co-cultures, repeatedly wash the THP1 cells in the plate and slide as described previously. Wash the cells at least 5 times to remove any non-internalized promastigotes. Then, add 100 microliters of 2% FPS RPMI and 100 microliters of each drug solution to each well or chamber, and incubate the plate and slide at 37 degrees Celsius for 48 hours. Following the drug treatment, wash the chamber slide 3 times in serum-free medium. Peel off the plastic chambers from the slide and fix the cells by immersing the slide in methanol for 30 seconds. Place the slide in a biohazard hood with laminar flow to allow it to dry. In the meantime, prepare a 5x solution of cyber green. Once the slide is dry, immerse it in cyber green to stain the cellular and parasitic DNA. Incubate the sample in the dark for 15 minutes at room temperature. After the staining step, wash the slide with water and let it dry. Using mounting medium, place a glass cover slip over the slide, then observe the samples using a fluorescent microscope and capture images of each chamber. In uninfected cells, only the macrophage nucleus is visible. In infected cells, the parasite nuclei and kinetoplasts can be seen in the cellular cytoplasm, attesting to the presence of intracellular L. donovani parasites. To quantify the infection, use ImageJ to calculate the percent infectivity, which is the number of amastigote nuclei times 100 over the number of macrophage nuclei. For the parasite rescue assay, first wash the 96-well plate co-culture 3 times with serum-free RPMI medium. To lyse the cells, add 20 microliters of 0.05% STS RPMI to each well. Shake the plate for 30 seconds, then add 180 microliters of 10% FPS RPMI per well. The time for STS treatment should not exceed 30 seconds to obtain controlled lysis of the macrophage without any effect on viability of rescued amastigotes. Incubate the plate at 26 degrees Celsius for 48 hours to allow transformation and proliferation of the rescued visible amastigotes into promastigotes. To quantify the promastigotes, add 10 microliters of Alamar Blue to each well of the 96-well plate and incubate it at 26 degrees Celsius overnight. Following this incubation, quantify the live parasites by reading the plate in a standard fluorescence fluorimeter. In the absence of drugs, the percentage infection increases with the number of amastigotes present relative to the number of macrophages. In addition, the quantity of surviving parasites determined by rescue assay correlates well with the percentage infection. In the presence of increasing concentrations of amphotericin B, the number of rescued parasites decreases in a dose-dependent manner. The parasite rescue and transformation assay may be summarized with sequential microscopic views. This picture shows intact THP1 cells in culture infected with Leishmania A mastigotes. These infected THP1 cells are subjected to controlled lysis to rescue live A mastigotes. The live A mastigotes rescued from the infected THP1 cells transform in culture into promastigotes. These transformed promastigotes grow and proliferate in culture. The overall number of promastigotes in culture is directly proportional to the number of live intracellular omastigotes.
This assay has been optimized for the controlled lysis of Leishmania infected THP1 cells. The objective was to optimize the conditions for detergent treatment, which yield almost complete lysis of THP1 cells with minimal effect on the viability of the rescued amastigotes. This step is very critical in this assay.
The parasite rescue assay demonstrated here can also be applied for evaluating infectivity of different species of Leishmania parasites causing cutaneous or mucocutaneous leishmaniasis in different macrophage cell lines, and also different Leishmania field isolates or genetically manipulated parasite cell lines. Both parasite rescue assay and image analysis assay show comparable results. They have the potential for automation and application for large-scale screening. We have successfully applied this assay for screening more than 16,000 fractions prepared by high-throughput fractionation of natural product extracts available at our center.
We have also optimized the image analysis assay by combining the images captured under DIC and fluorescence filters and quantification of the infection with ImageJ. ImageJ is a publicly available Java-based image processing program originally developed at the NIH.
Through this assay, several natural product fractions with novel compounds have been identified as new leads for anti-Leishmania drug discovery.